and the Deputy Director of the Institute for Integrative Biosystems Research and Education at Vanderbilt. He received his PhD from George Washington University and then was a postdoctoral fellow at Forschungszentrum Jülich in Germany with Professor Sabine Becker, and then a postdoctoral fellow at Texas A&M with Professor David Russell before joining the faculty at Vanderbilt as an assistant professor in 2006. He and his colleagues focus on the conceptualization, design, and construction of structural mass spectrometers, specifically targeting complex samples in systems, synthetic and chemical and biology. His group applies these strategies to forefront translational research areas in drug discovery, precision medicine, and human-on-chip synthetic biology platforms. He served on the board of directors for the American Society for Mass Spectrometry, and he serves in an editorial role on the boards of several leading scientific journals. He's received many professional and teaching awards including his laboratory being designated as a Water Center of Innovation, which I have to say, that's how I met Dr. McLean, because my husband works for Waters and said, oh, he'd be a great speaker. You should talk to him. And he graciously agreed to present to us today. He's published over 150 papers and holds 30 patents in the areas of innovative bioanalytics and quantitative sciences. And we're so excited to have him here today.
That was super kind. I have to tell you, it is really a privilege and an honor to be here. And I have been really excited about this particular talk for the past 2 months. And my kids were really excited. So I have an 11, a 10, and a 13-year-old. And because I am like dad joke extraordinaire, I have to say, so the 10-year-old really wanted to watch this new Disney movie last night. So we did Jungle Cruise. And if you really love really bad puns, it is absolutely fantastic. And The Rock did a wonderful job. So my daughter likes to say, you know, what if people don't laugh at your chemistry jokes? What do you do? Well, you just keep going until you get a reaction. So, okay, sorry. Had to start out somewhere. So let me start sharing my slides. Okay. So Kristen, if you can just give me a thumbs up that
we're good to go. I can see you on my screen just fine.
Okay, excellent. And I also, just at the outset, I shared my email address in the chat. And if you have any questions, I'll stay for the discussion afterwards too. If you want to stay in the discussion chat here, and I'd be also happy to share some of the experiences that we found in Nashville, either in our public school system or also at the university. And you know, I think everybody's been struggling. And to echo, you know, Mary's comments, you know, I commend all of you. Like, it has just been so difficult because of the emotion, the anxiety, just, you know, my own personal fear is everyone has been through a pretty major trauma globally. And so paying attention to mental health and well-being issues, both amongst our educators, but also our students, I think is really important. And also to Kathy's comment, I think one of the really cool aspects, at least that my students have gained from this, is with our scientific conferences and conferences like this, professional conferences, is it allows us to be really inclusive. And so, you know, individuals that may not have the financial means to travel to the conferences, either nationally or internationally, and also just being able to get individuals like some speakers that we've wanted from overseas that we haven't been able to get to the university for years, finally agreed because they could do it out of the comfort of their kitchen. And so, you know, so I think we have learned a couple of silver linings, but I tend to be sort of a silver lining individual. Now, I'd like to share with you sort of some of the philosophy of the research that we do. So I'm an analytical chemist, and I sort of liken analytical chemistry to be sort of the central measurement science, and all branches of chemistry sort of build upon our ability to measure things, the properties of atoms and molecules and, biology. And so I come from a family which is really full of educators. So my mother and father-in-law were both principals at public high schools in upstate New York. My mother was 7th grade English. And my father was really inspirational for the background here. So my father worked 3rd shift at the post office as a mail sorter. And later in life, he decided he wanted to also become a mountain climber. And that was a little out of the blue, but I would ask him, he would train for months on end so that he would climb these massive mountains with names like Aconcagua and Denali, and he's climbed mountains in Nepal. And I'd ask him, what really drives you to do these things? And he said, it's really fantastic when you get to the peak of where you're going, and then you can look around and see the journey that brought you there. And so in all of our research, we really sort of use that as inspiration. And if we're going to talk, a little bit about exploration in that context, I also want to turn, to some of the early biological explorers. And so here's Charles Darwin, and he's well noted for this quote: "It's not the strongest of the species that survives, nor the most intelligent, but the one most responsive to change." And what I'm going to do today is tell you a story about biology and medicine, and tell you really sort of where are the forefronts, and where, based on human knowledge, are we really pushing deeply. Now, this quote also says, not only do we need to map biology and health and medicine, but what we also have to do is look at time and how that biology and health change as a function of time. That places enormous demands on our ability to measure molecules. Now, if you think about sort of what does the future hold, and I think there are 3 really big areas that we're really making inroads today. And when I talk about things like big biology, I sort of liken them to what I would consider big physics projects. And so, like, by big physics project, I mean the Manhattan Project, right? Where if you bring enormous resources to bear on one singular question, what can you do? And I think biology is really well poised for this. And so we have things like the Cancer Moonshot and others that are ongoing. But in these 3 areas, systems, synthetic, and chemical biology, I'm going to tell you short vignettes about each of these. But in systems biology, it's really, how can we map you? How can we map on a personalized basis all of your biology so that we can do some prediction about health and how we can treat you asymptomatically, pre-symptomatically, for diseases that you may develop 5-10 years down the road. Synthetic biology is really how can we use advances to build biology from the ground up, things like human organs, or being able to use bacteria. I'll tell you at the end how to use that to make drugs and other commodity chemicals. And chemical biology is really how do we use our chemistry knowledge to inform drug discovery. Now, I wanted to contextualize some of the measurements that we're making now against really one of the most successful government-run biology projects ever. Now, all of you are familiar with the Human Genome Project. And it was really interesting from a couple of standpoints. So this slide is one that I took. I taught a class in the spring semester called, appropriately enough, Contagion. And I co-taught it with an immunologist and a drug developer. And so, that's sort of like the beginning of a really bad joke. So like an immunologist and a chemist walk into a bar, right? But it was really centered around informing our students of around the SARS-CoV-2 outbreak. Now, as part of that, we discussed the Human Genome Project so that we could go into concepts like PCR. But, you know, put yourself in the mid-1980s when this was being considered. So we knew that we have about 2.5 billion bases. And at that time, sequencing rates, we knew that we could sequence it, but it would take about 2,000 years. And so, you know, in Washington, they convened a committee of 8 biologists and 8 chemists, analytical chemists, and said, you know, should we do this? Because this is going to cost more than any one funding agency can do. And, you know, from an administrative point of view, you never bring an even number of people into a decision. You always bring an odd number because they split 8 and 8. So the chemist said, yes, let's do it. And the biologist said, no, let's not. And the chemist said, the reason why we think you should do it is you should fund measurements and get our sequencing rates up quickly. And then we'll bring down that 2,000 years, you know, pretty fast. So on the right side of this slide, you can see you and 130 volumes arranged by color of your chromosome, right? And if you want some reading that will put you to sleep, just start looking through gene sequences in one of these tomes, right? And so obviously we finished the first 2 copies, you know, by about 2001. So, you know, that's just incredible because they took the advice of the chemists. Now, to contextualize where that is today, you know, at the time the Human Genome Project was completed, it cost about $100 million to sequence a genome. By today's standards, you can do it for less than about $1,000, right? And so sort of that training in measurement science is what led to all of this. And I firmly believe being able to do that is good. And so here's Moore's Law, and more on Moore's Law in a bit. But now let's circle back for a minute, because if that's true, right? So we can sequence these genomes and we can do these things. Now, at the outset of the Genome Project, we thought that if we could sequence one human genome, we would understand just about everything we needed around a variety of diseases and everything. And in large part, what it did is it was a beautiful thing. It opened up Pandora's box and it showed us everything that we don't know. And so it was one of those cases where the answer to your question raised so many more questions that were unanticipated, which is wonderful, right? Now, when we talk about large mapping experiments, though, obviously this is the same organism, right? Same genome, but it has 2 different phenomenological or phenotypes as a function of time, a caterpillar or a butterfly. And so what's really driving measurements right now is being able to develop things like a Rosetta Stone to translate what is the biological transition from one phenotype to another by mapping all of the molecules, all the proteins, small molecules, and so forth, instead of just looking at the DNA. The DNA is important, but everything else is too. Now, this is not a new idea. So in the 1950s, there were physicians like Waddington that were already conceptualizing some pretty advanced ideas. So looking at the so-called epigenetic landscape, or changes to your DNA. And the way that he would liken the epigenetic landscape is as sort of like a topological map for a mountain, right? And so you would have these curves and these valleys, and your life trajectory would follow whatever that contour was. And since this is the association in New England, think of a skier, right? What is the potential energy surface that your life would take? And what was interesting is he already started developing these ideas of being able to change your topology through things like what you're exposed to, the drugs you've taken, the infections you've had, and what are your social traits. So how can we map out what those topological life maps look like is tantamount to what we're trying to do here. Okay. Now, I showed you sort of this quote from Darwin, and so on the left here is sort of a flux map. And so not only do we need to have the spatial coordinates of where those molecules are, we also need to know where they're moving as a function of time. And so looking at flux, molecular flux, both at a cell, but also at a tissue and organism level is equally as important. And so we sort of try to organize these things. This is just on the right is a snapshot of one piece of the metabolic map. But these maps are also incomplete. So it's estimated that even in simple metabolic maps, we're missing about 20% of the small molecules that are really key and important here. So we need to map space and time. So what does that mean for us? Well, personally, and speaking for myself, I view it as job security. But in about, let's say, I used to say 50 years, and now what I would say is about 10 years. If I was a student today, I would be so profoundly excited because our ability to make these kinds of measurements is just so unsurpassed. And it leads to a different problem that we'll talk about shortly. But what might the future hold? If you go to your physician, what you might say is, we're gonna run a systems biology experiment, map your biology. It might be blood, it might be tissue or something else. And then, the next day she'll call you up and say, I need you to go down to CVS or Walgreens, where we called in your personalized drug. And what we're going to do is change your wiring a little bit because you may have developed diabetes in 10 years down the road, right? But we're just going to nip that in the bud today. And so, there are a large number of investments and companies that are really moving quickly in this precision medicine approach. Okay, so now we're going to move to chemistry and analytical chemistry. Now, what do we need to create a map like this, right? And so, the Human Genome Project was up here, so about 20,000 genes. Now, when I took biochemistry for the first time, I like to consider myself young. I was still told that one gene equaled one gene product. And today we know that it's actually between 5 to 20 gene products, right? So that's transcripts. Now, proteins, you have something over about 100,000 different proteins, maybe 50,000 metabolites. But there's a lot of effort going into this bottom one here right now, the so-called exposome, or everything you've been exposed to chemically over the course of your life. Now, that ranges in estimates from 10 to the 8th to 10 to the 50th. All right. I'm a fairly simple individual, and I have a very difficult time, as well as many students, in understanding what does 10 to the 50th mean, right? Like, that's a really big number. So let's put some of these big numbers in perspective, right? So this year, we're a little bit over about 7.8, knocking on 7.9 billion humans on Earth. There have been about an estimated 150 billions over the course of human involvement on the face of the planet. I know this is early in the morning, so I apologize for this, but about 30 trillion cells in the human body. And as many of you may know, there are about 40 trillion bacteria in you and on you. And we'll get more to the importance of washing your hands later in the talk. Okay, now this number here is really pretty cool. This is the estimated number of grains of sand covering all of the oceans and all of the deserts across the face of the planet. And now this is the number of different molecules that we may need to be sensitive to measuring, right? I mean, that is ridiculous. And so that's where job security comes in. Now, we are making huge inroads in being able to sort of chip away zeros at a time, right? And that's what I really want to sort of share with you today. Okay. Now, one of the big problems with, well, it's not a problem, but just as an educator, when you think of like the size of human knowledge, people can make an estimate of this. It doubles every 2 years. And so when we're teaching students today about what their future may need, what we're really doing is we're projecting what we think they may need. And so if we teach them tools like critical thinking and things and prepare them for the unknown, I think that's a much better approach, right? Now, this is a really cool slide that was put together by Jody May, who Jody and I have worked together for several decades, and he's just a really deep thinker. So he was trying to measure what is knowledge expansion across time. And he turned to Buckminster Fuller, who was, Buckminster Fullerene is named for him, right? You know, buckyball, the soccer ball of carbon, right? Now, what's not really widely publicized is they think of him more as an architect, but he had hundreds of patents, and he considered himself as a futurist. So this first one is Buckminster Fuller. He was looking at some of these things, and he used the rate of element discovery as a proxy for knowledge. And each one of these red bars is a doubling rate, right? So, you know, it took us about 2,000 years to get to from about 10 to about 20 elements. And you can see that the rate of these elements being discovered just keeps going up. Even in my modest state of Tennessee, a few years ago, we had Tennessee, and you know, in honor of Vanderbilt and Oak Ridge, mostly Oak Ridge. Now, in the mid B is showing the rate of new chemicals being registered. C is the one that just really is daunting to me. This is why nobody can keep up with the literature any longer, right? Because it's the doubling rate for literature expansion. Now, on the bottom are a lot of semiconductor laws that, just to give you context about how our computers have increased in their capacities. And so based on this, what we really need to do is we really need to have a means by which we can organize really big numbers and really big data sets. And so what I'm going to do is tell you how we use things like artificial intelligence and machine learning to organize big data so that we can make sense of what it is that we're measuring if we're going to be measuring these massive numbers of molecules. Okay. Now, I'm not going to spend too much time here, but I do need to kind of contextualize what has really sort of been pushed after the Human Genome Project. And it's really mass spectrometry. And now in mass spectrometry, what we're doing is we're using electrostatics and magnetic fields in high vacuum to be able to measure the exact mass of a molecule very quickly. And it's the quickly part, which is why it has become so important. So over the course of millionths of a second or microseconds, we can measure the masses of all of the different molecules in a given sample. Now, I'll show you in a moment that knowing the mass does not solve what molecule it is. And what I mean is I can tell you the chemical formula very quickly, but there are so many different molecules based on their connectivity and their bond arrangement that could give rise to the exact same chemical formula, and that's problematic. So that's where this ion mobility part comes in. Now, all of you, if you have ever flown on a plane before, have seen an ion mobility spectrometer. So in all TSA security checkpoints, they have ion mobility spectrometers, which are not mass spectrometers. And what they do is if they swab a duffel bag or something, they'll put it to a device where they ionize the sample, and then they pass the molecule through a gas, an inert gas, usually nitrogen, where the gas experiences all these different collisions, millions of collisions, and then you detect it. And so what you're doing is you're counting how long does it take a given molecule to traverse this sort of thick, viscous gas. And what you're measuring is you're measuring what is the surface area of the molecule. And so based on that measurement, we know something about the molecular size. And so, you know, adding that to a couple other measurements, they can rapidly tell you whether or not you have explosive vapor on the cotton ball, okay? Now, the beauty here is because we're in the gas phase as opposed to the condensed phase, even ion mobility separations are done over the order of milliseconds. So in an airport, they can measure in less than a second whether or not you have, with exquisite sensitivity, whether or not you have explosive vapor or not. And what it does is it provides what we call a drift time. And so this scale here is milliseconds. This scale here is microseconds. And all of these dots are all of the different molecules that you get out of that given sample. So the reason why mass spectrometry has sort of become this darling after the Human Genome Project is because what we want to do is we want to measure everything. Okay, now this is one of my students. I was really flattered because he was featured on the cover of CNN News talking about some of these. But the spectra on the right is what this data really looks like off the instruments that we help develop. Now, we still do liquid chromatography to help give us more separation because we need 10 to the 50th, right? So each one of these gray dots represents a different molecule. So this is hydropathy, or what are its feelings about water using reverse phase or normal phase. Drift time is its structure, its surface area, and mass is its chemical formula. What is its mass? Now we can generate one of these plots where we can fit about 1 million molecules per second. Okay. Now we're talking speed, because if we have to get to these big numbers, we have to do it fast. The problem again is we generate a terabyte of data every 10 minutes. And so what do you do with a terabyte of data every 10 minutes, especially when you got 10 or 20 of these instruments going constantly, right? And so it places huge demands on informatics and biostatistics. We actually team up a lot with astronomers who deal with big data. And so we actually call these asteroid plots just so that they feel more comfortable with what we're doing. And so when they do star mapping, we do that now in biology for all of these molecular determinations. Now, it is profoundly important that students understand how to interpret spectra by hand, right? And we heard about drawing and things, and that is absolutely essential to know what is happening in the background, right? But we can't ask students to interpret all of their spectra any longer, and here's why. The way these kinds of measurements used to be done is by LC, and maybe with a UV detector. And so let's say we have an LC trace, took us 15 minutes, and we get one chromatogram with some number of peaks, right? Well, if we stick that on a quadrupole mass spectrometer, we could generate about 10,000 spectra across that elution profile. But by the instrument standards we're using today, this is a really conservative number. I have about 10 million spectra in those 15 minutes. The real number is somewhere around 50 million plus spectra. And then we also fragment them and take another spectra. So let's say 100 million spectra every 15 minutes. So you can't sort of hand annotate these. And so where we're seeing like the really great thinkers is where they're marrying computer science with mathematics, with chemistry, with biology. And it's really that intersection of these different disciplines which is really providing huge inroads. Okay, now I've already kind of mentioned this, but this gives you a sense of why we need the ion mobility part. And it's this so-called isomer problem. One chemical formula gives rise to a large number of different molecules. And so this is maybe about 6 billion compounds have been registered in PubChem. But even if we look with finer and finer mass resolving power, that is, how well do we know the chemical formula? Even once you get down to 1 part per million, I have this one chemical formula here that's going to give rise to 10,000 different molecules, just based on the molecular connectivity. So even just knowing the chemical formula is just not enough. And so that's why we need things like structure and feelings about water and so forth, so that we can really pin down which of those 10,000 are the important ones. Okay. Now, the other thing which was really kind of serendipitous, and it was not exactly predicted, was ion mobility, or separations on the basis of structure, gave us something which was really interesting. I'm sure you've heard the word omics again and again and again and again. And a good friend at Yale refers to omics, whenever you use it as a suffix, to stand for only massive infusions of cash will suffice. And the reason is because the scale of the experiments are so big that you know you're either going to be making a lot of them or the instrumentation is very expensive. But what we found is that this mobility by size or shape allows us to do multi-omics, or things like lipidomics, measuring the lipids at the same time as the proteins, at the same time as the carbohydrates. Because at a given chemical formula, those different types of molecules adopt very different structures. And so we could separate them all very quickly. Now on the right, if you zoom in on, say, lipids, you even find different classes of lipids separate really quickly into different types of lipid species. And so we've built things like crowdsource chemistry tools to be able to use these collision cross sections, or size, to annotate the different molecules that you're getting out of these experiments. And what I want to say is that, while this is being wildly adopted, globally, we're really at the beginning stages of being able to map the biology using things like size and chemical formula. Okay, and this is just sort of underscoring the idea that you can map different aspects of these isomers using the size measurement. And so this is a page out of what we call the lipid atlas, which is mapping, in this case, phosphatidylethanolamines, but we've done it from cerebrosides, a large number of different classes of molecules. But based on the size, you get different folding patterns based on the numbers of double bonds and carbon atoms in the fatty acyl chains. Okay, now what I'd like to do is I'd like to tell you two small vignettes around synthetic biology and chemical biology about being able to map molecules really quickly. Now, this first one was really a strange bedfellows collaboration between the Food and Drug Administration and the Department of Defense, DARPA in particular, so the Defense Advanced Research Project Agency. And we've worked with both for many, many years. So about 15 years ago, the FDA recognized that drug development costs, we all know that it costs billions of dollars to come out with the next drug. About 70% of that was just inefficiency in the drug discovery pipeline. And what I mean by that is when you're doing drug discovery, you start by testing your new molecule in vitro, like in cell culture, in vitro. And then what you do is if it looks promising, you then take it into an animal model. And if it passes all of the tests there, you take it to a human clinical trial. Now, the reason for that 70% inefficiency is you lose about 30 to 35% of your drugs when you move in vitro to your animal, let's say a rabbit or something, because the human physiology is different from the rabbit. You're not a rabbit. You're not a rat. You're not a mouse. And then so you lose those drugs and they never make it to the humans, even though they would have worked, right? Now you lose another 35% because of the converse of that statement. And that is, it works in the rabbit, but then it doesn't work in the human. All right. So the idea was, could we make three-dimensional human organs to remove animal testing from the drug discovery pipeline? And so for the past 15 or so years, we've been working very heavily, in particular with groups at Harvard, at the Wyss Institute, but also places like Los Alamos National Lab and others, in building large-scale, basically synthetic humans on chip. And so I'd like to tell you a little bit about that. Now, one of the things that you need to do, though, is deal with the information problem, right? And so how are we going to build the map when we do the synthetic biology? That is, how does your liver react? How does your kidneys react to a different drug or a toxin? Now, the image on the left is a famous picture from Fritz Kahn, who was a German physician in the 1920s, a Jewish German physician, largely credited with being the pioneer of infographics. And what Fritz would do is he would take really complex biology and he would create pictures to kind of make the general public understand what was happening with their bodies. And so you have sort of manufacturing plants and so forth and what's happening, you have computing going on in your brain. And so one is we need to reduce all of that data into an easily readable format. Now the other is really a concept which was put forth by Lewis Carroll, who's most famous for writing Alice in Wonderland. Right? Now, he did that with the president of Oxford University, who is a mathematician. And this particular quote really sort of sums up the philosophy of what we need. Is the Cartographers Guild struck a map of the empire whose size was that of the empire, and which coincided point for point with it. So basically, this is a map taking all of the information which had a scale bar of 1 mile equaled 1 mile. So this was a massive map that covered the empire, right? In fact, it went over your home and everything else. And the idea here is we don't need a map like that. What we need is we need to very quickly figure out which were the important molecules out of those millions of molecules that we're measuring that told us something about the important biology that was happening, either as a diagnostic or the prevailing biology. So what we do is we turn to internet commerce, right? And so we use a lot of tools that were developed by places like Amazon, YouTube, and others. Artificial intelligence. And so really a lot of the tools that we use could really be broken down to what you call a sliding puzzle game. So these are, being able to slide the different tiles to make a picture. So how does internet commerce use this? So when Amazon wants to give you a book recommendation, right? Or what do I think as Amazon you're going to want to buy next? What I'm going to do is monitor your page views, your buying history, your mouse overs, and so forth. And I'm going to make a picture of what that looks like. Now what I'm going to do is say, well, I have 400 million customers. So I'm going to make a tile for each one. And so they do. And then you slide them around and you figure out which two customers most resemble each other, right? Is it Kristen and Chris? And then what you'll do is you'll say, okay, I'm going to tell each of them to do what the other one did last as a prediction of what the other person would like to do in the future. And we do the exact same thing, but what we do are we make molecular measurements as a function of time. We create a tile for each molecule and we slide them around. Which molecules behave similarly to which molecules? Okay. So the reason for analytical chemistry is as we start building these different organs, and we use microfluidic chip devices to do that, we have to be able to measure very rapidly all of the molecules that the different organs are secreting at each one of these different junction points. So along the venous system and the arterial system. Now, initially we did a lot of work with a really great machine learning group at Cornell University and a group in physics building fluidic devices, but where we would use these to trap cells, different cells of the immune system, and then we tickle the immune cells with different drugs. We would then measure all of the different drugs very rapidly over the course of about a second, and we would feed them over to Cornell, where they would create partial differential equations to see how did those different molecules interact with one another. And so this is leukocytes, which are just falling into these little buckets like Plinko chips, like on The Price is Right or something, right? And then what you would do is you would put like a toxin or something like, we did experiments with cocaine for a while. And so you could see the immune system beginning to recognize that it had seen cocaine before. Right? Now, Cornell would then tell us what's the next experiment we want to perform. And this is what I would say is sort of modern analytical chemistry. So this is how do you build, for example, if you need to change the cell culture media for one of these organs really quickly, how do you make a 500 nanoliter solution every 5 minutes, but make it with custom ingredients, right? And so we turn to microfluidics. And so this is a design that we borrowed from Caltech where you have 32 different inputs. So this might be like sodium chloride, this one might be glucose, this one might be, whatever. And then it would mix all of these things. And then we would pump these back into the chip with a different organ. And we would build these up on 96 well plate formats where you can have different sort of perfusion control for the liquids. You would have different computer control and so forth. Now, I'll tell you just a couple of slides about a liver on a chip. And so this particular chip was built in collaboration with Charité Hospital in Berlin and at Los Alamos. And what would happen in Berlin is they would build what they called liver bioreactors, which are about 2 liters in size. If you were awaiting a liver transplant, but they couldn't find a good liver donor, what they could do is hook you up onto one of these devices and keep you alive for another 2 weeks. And what these are they're hollow fibers where you would have gas, cell culture media, and then waste perfusing through the device. And you seed the reactor with cells from someone that recently died, so a fresh cadaver. And the liver cells spontaneously organize around these hollow fibers and create a functional human liver. So all we did is make it much, much smaller, right? And so I don't want to talk about this too much. There are a lot of acronyms, and I apologize. But the main idea here is if you take things like acetaminophen, which is the acronym APAP. So acetaminophen is Tylenol, and it very strongly affects your liver. Things like ibuprofen, which is ASA here. ASA affects your kidney. So we're using this as a negative control. Very rapidly measure all the molecules that come off when you put a little bit of acetaminophen into the cell culture media. And so it's like hitting your liver like a bell and then listening to how it rings molecularly, right? And so then we'll use artificial intelligence self-organizing maps, or SOM. And then once we organize all of that data, only at the very last step will we identify what those molecules were that changed very rapidly. And so this is what some of that data looks like. So in each one of these panels, there's about 30 to 50,000 different molecules. So the top row, we're making measurements every 60 seconds up to 24 hours when you put a little acetaminophen in. The bottom row are all the molecules that go down in concentration. The top are all that go up. Now, within the first 60 seconds, when your liver sees acetaminophen, profound things happen. You start seeing things like bile acid dysregulation, which is a hallmark of liver stress, right? And you see all of these small molecules start changing. So once you see what those little islands of red are, what we're doing is we're taking 50,000 molecules and reducing them down to about 100. And those are the most important 100 that are telling you about the biology that happened. Sort of going back to kind of the Lewis Carroll idea, right? Okay. Now I want to show you how to use some of those techniques, but in drug discovery. And I'll sort of wrap up with this short vignette here. So I promised you something about hand washing and bacteria. So Tasha Sturm is in the Southern California group for the American Society for Microbiology. She has an 8-year-old son. All right, 8-year-old son's running around playing outside like all 8-year-olds should do. The second he gets in the house, she goes, all right, stick your hand in this Jell-O. And it was agar, right? So he sticks his hand in the Jell-O before he washed them. And this is what she cultured. I mean, it is just nasty, right? So hand washing is absolutely critical. Now, on the right is sort of a new idea that a lot of companies are starting to adopt. And that is using bacteria as repositories for new drug molecules. So what we do is, I know we have some people on the line from Kentucky, which is awesome. So the state of Kentucky and Tennessee, we share this commonality that we have 10,000 caves. So what we do is we have people go down in the caves and we collect bacteria. So these bacteria have never seen humans before, sort of in the places they go. But now think about this for a moment. If you're a bacteria and you grow in a cave, you grow under constant light, humidity, temperature, and so forth, but you also grow in a very resource-poor environment. So you're evolving constantly to figure out how to keep all the resources for yourself. Basically, what you're doing is you're making chemical warfare agents to keep all the other bacteria away from your food. So that's what we do is we take these bacteria, we'll do the genome sequencing, and we can predict what are the new compounds that they might be able to make. But we need to tickle them to figure out how to get them to express those different gene clusters, so-called silent gene clusters. Now, the same tools I told you for the human on chip, we use here as well. So what we'll do is we'll take our unknown bacteria. So this was from a cave called Snail Shell Cave. Now, we take another bacteria we know a lot about, like Micrococcus luteus or E. coli or something, right? And now the real question is, what happens when you co-culture them? That is, you put them under the same evolutionary stress that you think that they develop this capacity to make these compounds. So what we'll do is we'll map out the 50,000 or so molecules in monoculture, 50,000 in monoculture. What are the new molecules they express when they have to fight? So here are what the plates look like. Now we use the machine learning or AI tools to do just that. So we use a simple algebraic expression here, right, to get these little islands of red. So all of the blue are just sort of like ATP and housekeeping stuff. The ones in red are the new drug molecules, the ones they express to keep the food for themselves. So we colloquially call this Fight Club, right? Now I know I just broke the first rule of Fight Club. Now what we're doing is we're kind of moving it into what we call imaging mass spectrometry. And instead of doing like individual cultures, we're going to take pictures by taking ion mobility mass spectra at every spatial location. So millions of pixels. So we get millions of images back of the bacteria fighting with one another. So for example, this is the bacteria we want to know something about. Here's E. coli. And if we start taking molecular images this way, you get these beautiful images where you can see this fighting or interaction region. And that's where they're trying to get the resources. And we can start isolating new antibiotic molecules and other species. But what's really cool about this is we're starting to ask really kind of simple questions like who says what, when, and where, and why, right? And so really chemical communication between different species that are interacting with one another. But what it really does is it's accelerating a lot of our drug discovery assays. So then what you can do is you take that new drug and you start using it to see, is it helpful for cancer? Is it helpful for other ailments that people may have? And so this just gives you a sense of what one experiment looks like. This is just, we get millions of images and they're all different chemicals, but in their spatial context, and we can do it as a function of time. So kind of working our way back to the beginning of the talk, we need to build a topological map, but we need to see how that map changes as a function of time. Now, as many of you know, Jennifer Doudna won the Nobel Prize this year for CRISPR. And now what we're doing is we're starting to tweak the bacteria. So we'll go in and we'll use CRISPR to knock out their circadian clock. So we make zombie bacteria. But we start tweaking the genetics of the different metabolic cycles so that those bacteria will just become chemical factories. So this is supported by the Department of Energy. And what they want to do is they want us to make fuel for your car, but they want you to be able to do it at home. So this is pond scum, cyanobacteria, like algae, right? So you put a little in a Ziploc bag and you tape it to your window. And then after a couple of days, you just pour your bag into your car and you drive away. And now the way to do that is you have to really optimize the bacteria to be able to really express whatever it is you need. And so what we're doing is we're using these mapping techniques where we're genetically editing different colonies of bacteria, but reading out what is their ability or their proclivity to make these different molecules very rapidly. And so, you know, these kinds of things, instead of like 10 years down the road, I really firmly believe this is just a few years down the road, right? And so, you know, I won't belabor the point, but you generate these beautiful images of the molecular context of what these things can do. And now with that, I'd like to conclude. And I have a huge number of individuals that have done really fantastic work. I have the privilege of being able to say what all of my colleagues have done, but really the true science and fundamentals are done by others. I've highlighted the work of several of our collaborators, and I would very much like to thank these institutes for financial support. I have to tell you, it's really been a privilege to speak with you today, and I'd be happy to answer any questions you may have.
I just want to say that I'm really happy we got to hear about the Bacterial Fight Club, because my husband has been talking about it he's like, oh, I hope he talks about the Bacterial Fight Club, and you did. So yay.
I'm glad I could yeah. It's really pretty interesting, yeah. Lots of new drugs that way.
Mort Hoffman?
Yeah, I mean, this is a fascinating subject. I had never experienced and thought about what people are doing, lurking behind those new laboratories. What impact do you think this new biology or new biochemistry or whatever you want to call it, these tremendous advances that are occurring very quickly, what impact must that have on the teaching of chemistry, right from the early start, perhaps in elementary school, middle school, certainly in high school, and then college and university level? I mean, it seems that things are moving very, very quickly in the scientific front. And if we don't carry on and keep up with this in the teaching mode, then the problem that you mentioned right at the very beginning, that the students that we have within a couple of years will be in areas perhaps of science that hadn't even been imagined. So where do we go from here?
That's a really good question. And that's something that we think about and struggle with all the time. I will say that most PhD programs have really changed over the past decade to become much more flexible, right? And so rather than, and it's sort of like the de-siloing of science, which I think may be a good thing. I think really where students will find themselves most integrated into what I would call the new biology is no one is working in isolation any longer. Like you don't have sort of like one group working by themselves. And like my own group, we have over 50 collaborations with groups around the country, right? And the reason for that is nobody can be an expert in everything. And so as you're training students sort of in chemistry, one of the things that I think is really helpful is integrating them in teams so that they're learning and they're able to communicate with one another effectively, right? And the questions are so complex that one individual can have the right expertise. And so when I go home at night, my colleagues want me to read books about electrodynamics and stuff so that I can build a better mass spectrometer, right? But I want them going home and reading books about cancer biology and the places where they're experts. I also think the ability to communicate effectively is, the pressure to do so is higher than it's ever been and will probably continue to be. And that is being able to present their work in a way which is accessible to people that are non-experts. Where they're experts in something else, but when you have sort of that team sort of philosophy, then that's where they're going to be most effective. And I'll tell you, many of the well, myself included, I also struggle with this too, is being able to kind of find those crossover points with other disciplines where we can communicate in such a way that it's understandable by everyone. I'm not sure if I really addressed it sort of head on, but those are just sort of some of the things that I know that we struggle with on a daily basis.
Thank you.
was another question in the chat about, from Pam, about, her comment was that most of the work that you described was automated. What's your perspective on lab science in education?
So that's a really great question. Lab science is absolutely critical. There's so much automation going on right now. And many of you may be familiar with the National Academy report a few years ago called the Rising Storm. And what the Rising Storm, to distill it, everyone here is familiar with it, is one is trying to engage students in STEM fields. But part of it is giving them the tools where we can remain economically vibrant and sort of continue sort of the trajectory that the United States has always had as being very innovative. There are very few training opportunities for students to be able to do things like instrument construction or to understand how the automation is really working. And the only way to do that is through fundamental lab science and really being hands-on. And so while things are becoming more and more automated, we work with a lot of companies, and it's not necessarily because we have the most brilliant ideas or anything like that. It's actually because the companies want access to the students, because the training opportunities and being able to have students that have the right skill sets for sort of the jobs of the future are really sort of far and few between. And all of that is practical knowledge. So like in my own group, when I'm bringing students in, I ask them like, if you like working on boats or cars, or you're just really curious about, when you were a kid, did you pull your toaster apart just to see how the wires went together? Like those kinds of things, I think should absolutely be fostered. And now I'm kind of speaking as a father, like, there was a time where I would feel really comfortable pulling apart my rotary phone, but I feel much less comfortable pulling apart my iPhone, right? And having the confidence that I can put it back together. In fact, I'm quite sure I would. But trying to have those experiences in the chemistry context is all lab-based. And I will share something, since this is a group of chemists, and I mean no offense to my biology colleagues. We run really big programs in chemical biology, right? And so this is sort of the interface of biology and chemistry. All of our students go through chemistry because we consider it to be the foundational subject. And that is to say that it's a lot easier to teach a student that understands chemistry, biology, than it is to teach a student that understands the biology, the chemistry. And so the fundamentals are absolutely key. And then sort of being able to reach out across these different fields, it has to be off a solid foundation in chemistry. And there is a reason why they call it the central science.
I hate to have to cut this short, but we have another set of sessions at 11. And so Chris is ready with some links for everyone. I want to thank Dr. McLean for joining us today and telling us these stories and really showing us these applications of mass spec that I never thought of, certainly when I was in grad school. Of course, I'm married to a mass spectrometrist, so we hear about this a lot in my house, but not everyone has that luxury, but you can borrow them anytime you want. And I really want to thank you for coming today and sharing your story, and just give a big round of applause.
Unmute us so we can clap.
I appreciate it. And I'll stick around for a while in this Zoom. I think that's where the link was for.
Yeah, this Zoom will continue. If you want to stay and continue a discussion of the keynote address, you'll stay right here. Chris, are you ready to post those other links?